Thank you for your kind introduction. It's my great pleasure to be here today and share a little bit with you of the research we've been doing in our lab, Bioorganic Chemistry of Nucleic Acids at the Institut Pasteur in Paris. And today the story will be about the new approach we developed, which is called next generation chemoenzymatic synthesis of oligonucleotides. In our lab, we do pretty much everything related to the synthesis of modified oligonucleotides. We start with nucleotides, and also phosphoramidites to use the building blocks for both chemical and enzymatic synthesis of oligos. So overall, we can do analysis, purification, and isolation of oligos of different length and complexities. As for the interactions, we study neuroactivities, which are based on nucleotides, oligonucleotides, and also related enzymes. And we work on development of innovative strategies towards modified oligonucleotides. And we've been trying to develop useful synthetic tools that are based on oligonucleotides to further apply them to relevant applications in chemical biology, therapeutics, and biotechnology. Speaking of applications, today we have a broad range of applications for modified oligonucleotides, starting from short therapeutic oligonucleotides. We already discussed it a lot today, and moving to even longer, thousands of nucleotides sequences for mRNA vaccines. Modified oligos, they can also serve as storage media for digital information, as molecular probes, biosensors, in nanotechnology, using DNA origami techniques, CRISPR, gene editing, and templates. There are many others, of course. When we speak of modified oligonucleotides, what we mean by that, of course, are chemical modifications that are possible in all three levels of nucleic acid structure, starting from phosphate backbone, that we already witnessed with presentation of Debashi, with simple phosphorylated modification, phosphoroditioates we saw today, triesters, amidites, and so on. Base modifications, of course, both natural and artificial and non-natural base modifications. And of course, on the sugar backbone level, where we have different locked sugar conformations and or simply 2' substituted sugars. And also, when we look at the sequence of oligo, we have the 5' and 3' ends that can be conjugated in various ways, starting from fluorophores, biotins for sensing, magnetic separation, GalNAcs, which we also heard many talks today, for the drug delivery, cholesterol, fatty acids, all to improve drug delivery and cellular uptake. And of course, you can introduce various functional groups to further functionalize the oligonucleotide with the cargo you want to attach to it. So overall, in therapeutic context, all of these chemical modification, they lead to the high in vivo stability, bioavailability, high target affinity and specificity, and of course, improved cellular uptake. Recent Nobel Prize in Physiology and Medicine is a great example to demonstrate the potential of chemical modifications that can enable something very useful, which was the effective mRNA vaccines against COVID. And then the following question is how we can introduce these chemical modifications into oligonucleotides. Today, we mainly use two common strategies. The first one is chemical synthesis, which is based on the use of modified phosphoramidite reagents on solid support synthesis via rounds of phosphoramidite couplings. And the second general strategy is, of course, the use of modified nucleoside triphosphates as a substrate for DNA or RNA polymerases in enzymatic extension of primers. Of course, both of these strategies, they have their advantages. Chemical synthesis today is highly automated. It can be scalable to kilogram scale. It is very high in flexibility because with chemical synthesis, you can introduce basically any type of chemical modification you want to modified oligo. However, it is hard to imagine the synthesis of long sequences over 150 nucleotides with this strategy because the yield is dropping gradually from step to step, and even more if you use modified phosphoramidite. And of course, it's not sustainable, high excess of coupling reagents, solvents, and so on. Enzymatic synthesis serves as an alternative for a long time. You can synthesize easily long sequences. It is more sustainable. Our yields are good. However, the biggest drawback with enzymatic synthesis is that if you charge the triphosphate with modifications, it works well with base modification. But if you charge, let's say, phosphorate chain, backbone, base modification, sugar, those triphosphates, they struggle to be good substrates for DNA polymerases, especially those suboptimal commercial DNA polymerases. So therefore, there's always the need to develop some alternative solutions to overcome the limitations of those two widely used methods. And in our lab, we propose such a solution, which we call next generation chemoenzymatic synthesis. And the idea behind is simply that we use chemical synthesis to synthesize the short-mer triphosphate and monophosphate substrates, which were further assembled together by using either DNA polymerases or DNA ligases to craft modified oligonucleotides of different lengths and complexities. So here is the general slide that represents our approach. We have three parts: the synthetic part, where we synthesize the short-mer substrates by using either DNA synthesizer, or we can use it simple manual synthesis, which is affordable for any lab of organic chemistry. And then we take these short-mers, either triphosphates or monophosphates, and then we test them as substrates for DNA polymerases and DNA ligases. And what is important, we test them with commercial DNA ligases polymerases that can be accessible to anyone. So the first part, I'll walk through all the three parts. The first part is the chemical synthesis. We use simple solid phase synthesis chemistry, where we just optimize the excess of phosphoramidite reagents. We removed all cut-in steps. You don't really need this in such a short-mer synthesis. And then we can do it either on DNA synthesizer, which is fast. However, you're a bit limited to the scale. And then we also build this in the manual setup, where we can go to 100 mix easily of the short-mer substrate. That was the good part. We can synthesize them, and then we move to the first part of our strategy, where we synthesize a small series of these triphosphates, and we started testing in primary extension reactions with two simple systems. First one is on complementary template that can accommodate only primers, enzymatically elongated the primer, and then the second system was where we used our modified primers along with natural nucleotides. And we were happy to see the full-length product formation confirmed by gel electrophoresis as well as LCMS. But the interesting point here that we observed the product formation only when we use the phosphorothioate linkages. And that's something that is most probably caused by the esterase activity of polymerases that are capable of cleaving the phosphorodiester linkages when you use classical phosphorodiester linkages instead of phosphorothioate. So we currently continue to work on this to figure out really the mechanism, and also we are trying to find some engineered polymerases that can accept both natural substrates and also those that are highly modified. The second part of the chemoenzymatic strategy that we also use the modified short-mer monophosphates as substrates for commercial DNA ligases. And in this case, we use pentamers that we ligate repetitively on the complementary DNA template. At the beginning, we screened several commercial DNA ligases, and we have found that T3 DNA ligase is shown in the best activity. We tested various chemical modifications, namely RNAs, 2' substituted sugars, LNAs, phosphorothioates, and also modified ones having all three types of modifications coming from short-mers. Nevertheless, if you look at the gel image, we have a lot of truncated products, which wasn't ideal for us. And then you have two solutions. You either go to enzyme engineering, or you try to optimize the reaction. And we simply undertook the optimization campaign, and without going really to enzyme engineering, we were able to push these reactions almost all to full-length product formation, which was very great news for us. And then we decided to move to the next step, which was to ligate 4 different pentamers on top of mixed template. And here with our protocols, we were able to demonstrate that we can do the stepwise ligation. We excluded all of the cross-ligation scenarios. Then we can either use the long or short primers or without primers. It works well. Then instead of 5 mers, you can go to longer sequences. We can perform mixed ligation of DNA, RNAs, DNA, LNAs, and other types of chemical modifications. So overall, we saw a lot of flexibility in this approach, and then we decided to move to something even more relevant, which is the antisense drugs, of course. And as a first example, we attempted the synthesis of foamiversant with this strategy. And we started somewhere at 30% conversion towards the foamiversant product, and then we were able to push it to full conversion by simply going to more crowded conditions using PEG. Also, additional metal cofactors added as well, especially manganese. It has a lot of activity, especially for those modifications. And then what was also interesting that here we were like using not the monophosphates that are classical substrates for DNA ligases, but monophosphorothioates. And right from the beginning, we mentioned with unoptimized conditions that we have only 20% conversion when we use monophosphorothioates, and it was already 80 with monophosphates. Saying that monophosphorothioates, they are less reactive, and nevertheless, we managed to push the reaction conditions to full conversion, but we're still trying to understand the mechanism, especially of the stereochemistry, which can open the full new window to stereoselective synthesis of antisense drugs. So long story short, we were able to successfully synthesize foamiversant with good isolated yields. Also, some conjugate with functional groups at 5' that you can use for post-functionalizations. We also did several examples with fatty acid conjugates. There are nice papers showing that these fatty acid conjugates that can form agglomerates and basically encapsulate the oligonucleotides inside, having these fatty acids hanging around, and then also the cholesterol analog for improved drug delivery. We are also happy to see that our approach is compatible to the synthesis of LNA gapmer designs, and here is successfully synthesized with exactly the same conversions and isolated yields for this foamiversant LNA gapmer. And it's very nice approach if you really go into design and you want to synthesize a diversity of and series of oligonucleotides, and you don't really need to resynthesize the whole sequence. You just manipulate the short fragments, and then you go with the synthesis of your series of antisense drugs. And then we went to another dimension, which is the synthesis of ultra-long modified oligos. And here we were able to successfully ligate 18 pentamer fragments on top of template. And the reaction works quite cleanly. We got our isolated products with diverse chemical modifications coming from those shortmers. And then we decided to combine this approach with where we ligated shortmers by ligating then these long mers that we form on the shorter template. And here we did several examples, and we can basically for the moment go to 300 nucleotides in length quite easily, which is something beyond the capacity of standard solid phase synthesis. That's it. I will just shortly summarize so that we really saw a lot of flexibility in our chemoenzymatic approach, and it has a lot of potential to be used in diverse applications. We are still trying to do more job on other types of ACOs, and then we are also actively using this approach in digital information storage project that we're running in our lab, where we do parallel synthesis of massive number of DNAs, and also these ultra long templates for that as well. And before I finish, we've been quite active on this field over recent years. We just published a nice comprehensive review on advances in biocatalytic and chemoenzymatic synthesis of oligos. So if you're interested, please check it out. And finally, I would like to thank all of my group members, especially our team leader, Marcel Ollenstein, all of our collaborators, funding support, and you for your kind attention.